The HubMap Data Portal features several ways to access, explore, visualize, and download datasets. The Vitesse viewer shown here is embedded into the data portal and allows rapid visualization of HubMap Common Pipeline process datasets. The navigation bar on the data portal provides access to datasets from different perspectives, such as the donor, sample, dataset, or collections view. The Atlas and Tools drop-down menu provides quick access to several important tools. The Common Coordinate Framework portal is a launch site for accessing the many 3D models and underlying tools and data required to build an atlas. The Anatomical Structures, Cell Types, and Biomarkers, or ASCT+B reporter visualization. The registration user interface allows researchers to register new tissue blocks from which data will be generated from a variety of different technologies. The exploration user interface for exploring data from the whole body and organ perspective, and to see registered tissue blocks within organs representing datasets on the portal for those organs. And the Azimuth reference-based single cell mapping tool allows users to map their data to existing organ reference maps and allows cross-species comparison to existing human reference maps. Each of these tools will be featured in the current VHMOOC modules, and we hope that you find these resources useful. And if you have any questions, please feel free to reach out to us at info.ccf@indiana.edu.
Hi, my name is John Hickey. I'm a postdoctoral fellow in Gary Nolan's lab at Stanford University. And today, in this video, I'm going to show how we can access some of the raw data in the HubMap portal, see what the metadata is, know how that same data, that raw data, can be processed by the comment analysis pipeline or downloaded and then processed via other pipelines. Explain how this data processing works at a very high level, and explain some of the caveats with working with codex multiplex imaging data. So first, we'll just go to the HubMap portal. And the way that I usually find that is by Googling HubMap Consortium. Go to hubmapconsortium.org. This is the main webpage for the consortia. It'll explain a lot of things of what's going on, where there are different centers, what kind of donors, events that are going on. Really useful place. But what we're going to go to today is the data portal, where all of this data has been uploaded, and we can see the different types of assays that are have already been uploaded, and this will continue to change as more assays and centers get involved. But what we do first when we go to this data portal is, if we want to look at a specific data set, we can click on the tab data sets. And then what pops up are all the data sets that have been uploaded to the portal so far. And we can filter based on data type, organ, specimen type, etc. So since we're interested in codex data, I'll just click on codex, and so that we can only see the codex data. So we can see that there have been a number of different codex runs uploaded for different organs, the large intestine, small intestine, spleen. So we're going to look at just the large intestine today. So to look at this data set, we just click on this HubMap ID. And this takes us to this data set kind of landing page. And here you'll be able to see some information about where this came from, some information about the donor, how old they were, race, and then also about metadata from the experiment, the codex experiment, what microscope it was used on, how many antibodies were used, who did it, the protocols that were used, the resolution. And then what you can do is then go to Globus to actually download all the raw files for this data set. And you would have to then go sign in through Globus to do this. And what you can do then with those raw raw files is, you can look at them, but they're going to be in a state that it's not easy to look at them or analyze them, because you have to collect many Z stacks for codecs to make sure you have the best focus folder, and they're not stitched together. And so what we have developed in the Nolan lab is a pipeline for dealing with this data. So first, what we do is we've developed a software, a codex uploader, it's called, that deals with tile stitching, drift compensation across multiple cycles of the fluorescent imaging, combining all these cycles into one image stack, doing background subtraction, and doing applying deconvolution to the imaging. And it comes up with the best focus Z stack from each tile, so that the whole image across multiple tiles has the best focus of that fluorescent imaging. So then once all of this data is processed, it's easier to look at ImageJ or other image viewing softwares, and to evaluate the staining, but then also to make a more quantitative assay of the data. We put it into a segmentation algorithm. So what this does is it uses the nuclear stain, which we have throughout the multi-cycle for drift compensation. So this aligns the images across cycles. But we have the nuclear stain also for segmentation. So we can segment out each individual nuclei based on this nuclear stain. There's also a possibility to use a membrane stain in combination with a nuclear stain, so that you can segment out also the membranes. And the segmentation tool that we also have as a software through our GitHub that I'll show you later is based on a watershed segmentation algorithm. And this then, after you apply this algorithm, it gives you mask around where the cell is. And this can be used to segment out each cell. And the reason you want segment out each cell is then you can then quantify the average fluorescent intensity for each cell. And this is what allows you to get the intensities for each cell and data, so that you have for each cell each fluorescent intensity. And this is similar to like flow cytometry like data, where you can have the fluorescent intensity for each marker for each cell, and you can plot that like we are here for this codex data, where you have CD3 versus cytokeratin. So cytokeratin an epithelial marker, CD3 a T cell marker. And you can see you can gate this similarly to how you do it with flow cytometry, where high expressing cells are likely the T cells, and high expressing of the cytokeratin are likely epithelial. But because this was captured spatially, we still have the spatial positions of each of these cells, which is represented by a single data point here. And we can look at where these cells are. So these stain, or not stain, but are present where we would expect epithelial cells to be. This is a tonsil tissue. And these T cells are around areas where we would expect the T cells to be. But besides just gating cells based on marker expression, usually codex multiplex imaging will have up to 60 markers. And so that's hard to do differential gating or hand gating. And so a lot of times what we will do to analyze the data to identify the distinct cell types is to do unsupervised cell clustering. So we also have another software that we've developed for this called Vortex, which uses X-shift clustering or k-means clustering. And basically you feed in your data that you got out of the segmentation tool, which is just like an FCS or a CSV file. You set the parameters you want, whether there's normalization to the data or not. And this then does the unsupervised clustering and outputs a number of clusters. And then you would go and annotate based on the expression profile and location in the tissue what each cell type is. So this is something that you can do if you download the data raw, you process using this processor, then segment, and then do unsupervised clustering to get cell types. And then I can show you some data of how that could be useful. So this is data that we have from the data that we've actually uploaded already to HubMap. And what we're doing at Stanford is looking at the small intestine and colon. And so what we do is we take 4 different sections from different areas of the small intestine and 4 different areas of the colon. We put them on the same cover slip, and we run a 44 antibody panel on those tissues using the Codex imaging technology. And you can see the types of regions that we ran. These are H&E stains after the Codex imaging. This is an example image of Codex multiplex imaging at 2 different zoomed in views. And here there are only 5 of the 44 markers shown, but it illustrates how good of resolution and how many markers you can do, and the types of cells, right? So CD3, you can see the red, and these are T cells, CD19, the B cells, vasculature, epithelial, extracellular matrix. So we can generate images like this after processing the data, but then we can go through and we can annotate these cell types. So for example, like this cluster, we are now looking at its expression profile, where CD31 is overrepresented here compared to other stainings, and we can label this as vasculature. And after going through all this, we can then have a percentage of cell types across the different areas of the colon or small intestine, and look at the cell types relationships to each other based on their marker expression, and also the expression of specific markers, especially ones that are maybe more phenotypic. So these are minimal spanning trees of these relationships, of these cell types that we've seen in our data. And then finally, because we have the XY positions, we can map these cell types back to the tissue images, as shown here, where the different colors represent different cell types that we found within these tissues. And further spatial biology-based statistics can be done, like neighborhood analysis at this point. So, but going back to, and I guess I should share that all these softwares are available on the Nolan Lab GitHub, and particularly the ones for processing Codex data. Both the Codex uploader and segmentation can be downloaded from our Codex page. So going back to this data set, so this is the raw data that we just explained, but the cool thing that HubMap and the Hive at HubMap is doing is generating a common pipeline that can analyze all imaging data sets. And so we can look at derived data sets. And what that means is we have this raw data set, but it has been put through this common pipeline that uses Cytokit. And we can click on this now. And we can then go in to Cytokit and look at the outputs that it creates. So it follows kind of a similar general strategy as our own derived pipelines, where it'll process the images and then do cell segmentation and marker quantification. So I'll just toggle on a few things, and an unfortunate thing that it doesn't do quite yet, but I'm sure is in the works, is stitching of all the tiles. So we usually take 63 tiles, so a 7:9 region at 20x resolution for each of the sections. And so right now in the viewing window, you just see one of these 63 tiles of one section. So it's not entirely helpful to just look at this, but what you can see is that the segmentation's already been done, and the mask are these colored features that are overlaid, and we can change a lot of features to look at the different cells and cell types on this tissue. So we can change the cell segmentation tools so that we only have the outlines here instead of just the full mass of what's being quantified. We can toggle over and look at each individual cell and what label each of them is, depending on the type of clustering you can do here. So I'm just going to turn this off and just show also a feature of where you can look at the actual marker expression. So if we want to look at like cytokeratin, we can look at that stain and we can adjust it for the brightness and contrast here to view this within this specific tile. You could look at an ECM marker. So we scroll through all these markers to collagen 4. We can see where that is expressed. We can also look at immune markers like CD45. And we can even toggle off markers if we're not interested in them. Oops. So you can see we can generate nice images and start to see where these cell types are. And then, if we want to see where cells are overlapped, we can do that. And what has been done here is an automatic k-means clustering, and that is the results generated down here where you have genes. In our case, they're not genes, they're proteins that are quantified by the antibody staining. And then the clusters that it identifies with the color corresponding to the colors here. So you can assume that this cluster is epithelial. You're seeing epithelial cells here, and they're all clustering in the same type, like since they have the same marker expression overlaid. So you can also download this data down here, and you can download that directly. You can see some of the outputs for each tile that it generates, some of the means. You can get the shapes, the clusters. These things can be used in your own analysis as well. So while this is a work in progress, it's already a good way to, if you don't want to process the data yourself but you want to look at some of this multiplex imaging data, you can come here and look at it processed through this common pipeline. So I'm just going to throw this up. We did a few of these simple things today in this video tutorial. I'm going to put these up here just at the very end. These are some helpful resources. So the portal website, our imaging software download from our GitHub, the manuscript that I walked through a couple of the figures, and my contact info. So thank you for joining me, and have a great day.
Hey everyone, my name is Andy, and in this tutorial we're going to have a look at the Exploration user interface originally developed for the Human Biomolecular Atlas program, or HubMap, but in this case ported over to the GTEx portal, as you can see here in front of you. Now, the challenge that we are addressing with implementing this user interface or porting it to the GTEx portal is that several common fund projects such as GTEx and HubMap, as well as others too, create large molecular and spatial data sets on human tissues. And of course, for this data to be interoperable and reusable, we need to accurately map and compare data from the same organs. Now, GTEx samples are collected across many individuals for the same tissue site using their standard operating procedures, or SOPs. while HapMap samples are collected for fewer individuals but for many specific sites. So the goal then is to spatially map samples collected under the GTEx SOPs to the so-called HapMap Common Coordinate Framework, to which these HapMap samples are already mapped. So this exploration user interface that we have here in front of us, or also called the EUI, is a way to semantically and spatially and also ontologically explore these data sets in their spatial context, in their semantic context. So when you get to the site, you can see that this is all web hosted. You will see that we have the top part here is GTEx, and then here we have the embedded exploration user interface. You will notice that you have this prompt down here that informs you that we log the usage to improve the service. We use this data only to improve the functionality of the interface. You may choose to opt out, or you may click that you understand, and that you understand. You can see that by default, currently in my view here, this interface is dark because I like dark UIs, but you can set it also to the light mode. By default, actually it's the light mode. So I will demonstrate the light mode here because that's what the default is. So the interface consists of three major parts. We have the left pane here with the filters. We have the 3D stage in the middle, and we have the details pane here on the right. Let's start with how to operate the 3D pane. This is a fully functional 3D window. And if I click and drag my left mouse button here, you can see that I can rotate my virtual camera around this assortment of reference organs here in the middle. If I press and hold my right mouse button, I can pan, so moving the camera sideways. And if I use my mouse scroll wheel or two fingers on the touchpad, if you're using a touchpad on a laptop, you can zoom. Currently, you can see that there's only a small selection of organs turned on by default, but all of these organs up here are available. So for example, we can turn on the large intestine, we can turn on the small intestine. Now you can see it gets a lot fuller in there. Let's go a little over here. We can also turn on the thymus. You can see these numbers here tell you how many tissue blocks are currently registered and available in the exploration user interface in here. Now I'm going to turn off some of these organs just because this is running in a web browser and we want to be respectful of the resources that we are using. So I'm going to turn off the thymus, the spleen, the small intestine also. I'm going to leave the skin on just for reference. We can turn off the large intestine as well. And let's turn off the kidneys here too. So now we only have the heart and the skin left. Now, in many instances, you may not want to see all of the tissue blocks that are available. So we have a set of filters available in the top left here. So if we click on this, we have this flyout. It allows us to choose a sex. Let's maybe go with male. We can define an age range and a BMI range, which I'm just going to leave at their default. We can choose different assay types or filter by different assay types, which is not currently super relevant for GTEx, but it is very relevant for HapMap. And then we can filter by tissue providers. So let's just choose GTEx here and click apply filters. So now we have our male reference body. You can see that we currently have 4 specific tissue blocks in here. You can see that we have 14 sites total. Okay, so but now what we see here are only 4, just for the heart and for the skin. So you can see here skin 1, 2. And then there are 2 more up here. One is in the atrial appendage, the other one is in the left ventricle. So now let's actually look at what we can do with these tissue blocks. So if I click on one of these, for example on this one, it'll tell me, okay, we have 293 donors currently for this particular site. You can see the mean age here is 56.4 for the males anyway, and the range is 20 to 70 years old. And then click here. You're actually going to open the GTEx portal, okay? And this is going to allow you to look at bulk gene expression and expression QTL data and explore all of that in more detail here on the GTEx portal itself. Now the interesting part, of course, is when you can compare data for the same site between HubMap and GTex. So in order to do that, I'm going to go to my filters. I'm just going to reset. Actually, let's just undo this one. Click Apply Filters. So now all of the other data, mostly HubMap data, comes just back in. And now, if I zoom in here a little more, we can see that there's actually an overlap between the tissue block for GTex and the tissue block for HubMap. So now, if I click on the HubMap tissue block, you can see that it is now colored orange. And then I can expand this here, and I can see who from HubMap registered it. And then if you click on this field here, you can inspect the donor information. This is the HubMap portal, just visible here inside of the Exploration user interface. Close that again. Then you can similarly look at tissue block information as well. Notice that you can also open this separately in a new tab by clicking this Open in Portal link up here. So it is, of course, interesting to have some more filtering capabilities. So here in the top left, we have a body partonomy where we can search for specific anatomical structures. For example, let's search for the septum. You can see that this is a controlled vocabulary. You can click the septum. And now the septum here is highlighted. And we can click it. And now we only see the tissue blocks that currently intersect with the septum. We can do that for all of the other anatomical structures in here as well. If I click it again, then the filter disappears. Now, likewise, I can do the same thing for cell types. And to make it a little more interesting, let's maybe turn on some of the other organs, also just to present you a little bit of a different view. Here we go. So now, in the cell type listing, we can see that, for example, for this cell type here, apocrine cells, we currently have 34 tissue blocks that are predicted to have this particular cell type. And we are predicting this via the anatomical structure, cell types plus biomarkers, or AST plus B table effort that is also part of HubMap. So then if I click this, now we can see that I have the 34 tissue blocks available here in the details pane on the right side, and these are predicted to have these cell types specifically. And then I can click on this again to reset this particular filter. All right, I want to give a little more information about some other elements of the user interface. For example, here in the top right, we have the circular arrow button that allows you to reset the view. It can be challenging sometimes navigating 3D spaces on 2D screens. That's why we included this. You can also click on this link up here and get to the CCF portal, where you can read up about the Common Coordinate Framework effort that is part of HubMap. We also, on the top right here, have an info button, where when you click it, you get access to demo materials, some about information, information about HubMap itself, information about the Common Coordinate Framework, or CCF, as we discussed before, with the same CCF portal link that you have up here, and finally, acknowledgements. One more thing that I would like to show that is kind of cool to look at is concerns the details pane over here. For this, I'm going to hone in on a skin tissue block. Let's go, for example, to this one back here on the back of the right arm. I'm going to click on this. And then we can see again, this is a HubMap tissue block. And this now shows us the number of tissue sections that are in this particular tissue block. This should help you explore all of the data that is cross-mapped here between GTex and HubMap in a little more detail. Now let's zoom out and admire these human bodies here again in their full beauty. So in this tutorial, we investigated the Exploration user interface embedded in the GTex portal. We hope that this helps you to cross-compare data from GTex and HubMap while they're all being mapped into the same spatial, semantic, and ontological space. Thank you very much.
What makes HubMap's registration user interface and exploration user interface able to handle a rich diversity of data from a broad variety of data collection methods? To answer that question, we need to understand how both programs leverage a common coordinate framework created specifically for the Human Reference Atlas, the CCF-HRA. The CCF-HRA is a computational framework that combines expert-curated ontologies for specimen, biological structure, and spatial data to create an underlying reference map of organs, tissues, and cells in the human body. The RUI and EUI use the CCF-HRA to describe collected tissue blocks and biopsies in a standardized way. This enables data harmonization across consortia, donors, organs, and assay types. As of May 2022, the CCF-HRA's 3 ontologies have been fully implemented for several organs, including the kidney, which is shown here. This enables the RUI and EUI to capture who, what, and where information for every tissue data sample. The WHO information, specimen donor metadata, is integrated and organized with the CCF-HRA specimen ontology. To create this specimen ontology, the more than 100 metadata fields covered by HubMap clinical data was reduced to a smaller set of fields most relevant for CCF-HRA construction and usage. The specimen data was then compared with data made available by other consortia to finalize the field selection. Currently, CCF-HRA specimen data includes demographics and clinical data, workflow information, author information, and links to source data. The what information is captured by the CCF-HRA biological structure ontology. While organs differ substantially in their form and function and across human specimens, all have anatomical structures, cell types, and biomarkers that are captured in the ASCT+B tables and the associated 3D reference object library. The resulting ASCT+B tables represent expert knowledge and information from textbooks, scholarly publications, and experimental data evidence. An associated 3D reference object library captures the anatomically correct 3D shapes, sizes, locations, and rotations of key AS present in the ASCT+B tables. These reference organs are developed by specialist 3D medical illustrators, and they are approved by organ experts. Finally, the CCF-HRA spatial ontology provides the framework for the where information. Spatial data describes the 2D and 3D shapes of entities and their physical size, shape, location, and rotation from the whole body level down to single cells. A unique feature of the CCF-HRA is the interlinkage of the ASCT+B to the 3D reference organs, which supports new forms of semantic and spatial queries. For example, given a registered tissue block, the 3D anatomical structures it contains or collides with can be determined and used as annotations. Using these AS annotations, all AS and CT inside of these can be retrieved. It is also possible to use cell types as search queries and to retrieve all AS that typically contain these CT.
Hello everybody and welcome to this demo of the RUI version 0.0.0. In this tutorial we're going to go over the functionality of the RUI, how to use it for spatially registering tissue blocks into reference organs in the Common Coordinate Framework. So as we go into the RUI, which as you can see is web deployed here, we are being greeted with this modal, meaning a window that we have to go through to get to the actual application. So we're prompted to type in our name. So I'm going to type in my name, first and last. Then we choose the donor sex. In this case, let's go with female. And then we have to select an organ. So we have this organ carousel here. Let's go over here to the lung, one of my favorite organs. Then we click Start Registration. And here we go. So this is the RUI proper. On the left hand, we have the so-called anatomical structures pane. In the middle, we have the 3D pane. And on the right side, we have the manipulation pane. The anatomical structures pane here on the left shows us the, well, anatomical structures that we have inside the 3D organ here. You can see that these parts here, they correspond to individual substructures in the big organ here in the middle. So for example, I can turn these on and off, and I can change the opacity. So you can see as I hover over these, you notice that this opacity slider here appears and disappears. So I'm going to go ahead and I hover over left lung hilus, and then I just play with this opacity slider here. Then you can see down here in this area, this has become more opaque now. Let me change that again for you, something lower. Here we go, that's almost completely disappeared. In fact, I can turn it off completely by clicking this eye icon. Let me turn on the opacity a little higher so you can see it better. So I can turn off, on, off. If I want to reverse it to the default, I just click the circular arrow button. And I can do the same thing for all of the anatomical structures using the circular arrow up here. In order to demonstrate landmarks, we're going to go over to another organ. We're going to go to the kidney. You can always change your reference organ using the carousel up here. I'm going to go to the left kidney. Here we go. So for the kidney, we have one landmark, which is the bisection line. So I'm going to hover over this. They are turned off by default. I turn them on, and I turn the opacity up. And you can see this green landmark being imposed here, and this is the bisection line. Okay, so much for the anatomical structures pane here. Let's go to the meat of the matter. Let's go to the 3D stage, the 3D pane. So here in the middle we have the organ. This is a 3D space, okay? And then you can also see that we have camera controls up here. So here I can change the virtual camera that I'm using to look at my organ. The organ is static. We are just moving a virtual camera around it. So here I can rotate around the organ in 90-degree increments. So currently I'm looking at it from the anterior perspective. And now when I click this button, I look at it from the right side. And if I click this button, I look at it from the back or the posterior perspective. In order to help us understand the orientation of our camera to the full body, we have this little mannequin here on the right. So you can see anterior looking from the front. Now we're going over to the right, and so we can go over to the left, etc., etc. Of course, this is very restrictive. So we added this 3D preview mode. So you can go from the rigid camera control, which is in the register mode, to a slightly more free version, going over to the 3D preview. And now I can use my mouse buttons. Okay, and I press and hold my left mouse button, I can rotate the camera. If I press and hold my right mouse button, I can pan the camera, so I can move it sideways and up. And if I use the mouse scroll wheel or two fingers on your touchpad, depending on how you're using your machine, you can zoom in and out. So for now, I'm going to go back to the registration mode. Okay, so you might have noticed this yellow tissue block over here. This is the tissue block that we are going to register. We're going to talk in a second about how you can resize it and rotate it. For now, let's just talk about how you can move it. So there's two ways for moving it. One, when you are in the registration mode, you can hover over it with your mouse and then press and hold the left mouse button, and that way you can move the block along 2 dimensions. Okay, so currently we are moving the block along the X and Y axis. You can see the coordinates of the block. Those are the coordinates of the block, going up and down, left and right. You can see that the Z axis is static. Okay, now if I switch to the right side, let's go to the left so we can actually see it, and I move it around now, you will see that the X axis is now static because we're moving it along the Z axis and along the Y axis. All right, let's get this back over here. Let's go into the anterior view. Now, a second way to move the tissue block is by using the Q, W, E, A, S, D buttons. Okay, so You might recognize this if you've ever played a video game on a keyboard. So if I press the W button, you're going to see that the tissue block now moves upwards in 0.5 millimeter increments. If I press S, it moves downwards. You can see how the Y position is decreased. Likewise, A and D moves it along the X-axis. Okay, again in 0.5 millimeter increments. And now let's go over to the right side to see the Z-axis movement. That is done with Q and E. So Q increases the Z position and E decreases the Z position. This is a fantastic way for making some really fine adjustments that you might not be able to do with your mouse. Now, the neat thing about this is that this also works in the 3D preview. In the 3D preview mode, we use our left mouse button to move the camera, so we can't move the block. So if we want to move the block now, we can simply use our keyboard. So let's say I wanted to align this tissue block with the bisection line. I can go back to register, go back to anterior, and now let's go over here to do the coarse alignment. Okay. And then I go to 3D preview and let's zoom in. It's a little far away. Let's go to the left side here. Actually, let's go to the right and move this closer. All right, now we're getting pretty close. Now let's go to 3D preview mode. Rotate around, zoom in, and pan slightly. All right, so now we're at the point where we have to rotate the cube. And all of this is done here in the manipulation pane. So let's go over this one by one. Here you can set the tissue block dimensions. You can see width, height, and depth. The default is 1 cubic centimeter or 10 cubic millimeters. I'm going to leave this as is for now. If I were to resize this, let's make this a little bit bigger. You would see that this reformats instantly. Let's go back to 10 millimeters. And one thing that I would like to emphasize here is that when you set the Z dimension, so when you set the depth, make sure that this aligns to the cutting direction of your cube. So for example, if you were working on a cube that you were dissecting into slices along the long side, you should make sure that the long side is going to be the depth. This is very important. So let's go back to 10 for now. So now the interesting part, how do we actually align this? Here, by the way, you can type in the thickness of the slices and the number, although that is just metadata right now. And then here we have the tissue block rotation. So you can use these sliders to rotate the tissue block around any of these 3 axes. Okay, so let's go ahead and try that. I want to align my block with the bisection line. So first I can rotate around the y-axis, like so. Looks about right. And now let's rotate around the x-axis. Here we go. It's always a little bit of a trial, but there we go. And now we can use our keyboard again to move the block farther towards the bisection line. I'm using the E key and then the S key to really get a nice and proper alignment without having to use my mouse. Beautiful. All right, now we are aligned. We are just on there on the bisection line. Of course, we are also somewhat close to the edge of the kidney, so let's move it around a little bit by using the D and the E button. Here we go. Now we're just moving towards the inside of the kidney. All right, wonderful. If you would like to reset any of these dimension, tissue slices, or tissue block rotation, you can also use those circular arrow buttons. So finally, I would like to talk about the anatomical structure tags dot here. You can see that this is quite populated. So to demonstrate this, I'm going to reset the rotation and the position of the tissue block. Here we go. So now the tissue block is outside of the kidney, and you can see here that the anatomical structure tags down here are completely unpopulated. Now, as I grab the tissue block and move it into the kidney, you're going to see suddenly a whole bunch of these pills appear. And you can see that all of these correspond to an anatomical structure over here. So we have the renal column, for example, which we have right here. We have the outer cortex of the kidney. Outer cortex of the kidney is right here. So we are using a feature called collision detection between 3D objects. If you've ever watched a Hollywood movie or played a video game that uses CGI, oftentimes collision detection is being used to compute physics that should happen afterwards, such as an explosion or deformation. Now, in our case, if two objects collide, it's something much more innocent. We just get an anatomical structure tag. Okay, so I'm gonna pull it out again. You can see that they disappear. This is all done in real time. Pull it back in. And then you can see that even as I move it inside of the kidney, these anatomical structure tags change because we constantly collide with new things as we go along. Okay, now let's say you have a tissue block here and you read these anatomical structure tags and you're like, well, it really does not collide. I did not take the sample from the capsule at all. You can just click this X here and delete it. And then you could, for example, say, well, but I think it really collided with the hilum of the kidney, but I don't see it tagged here. Then you can just type in hilum. And you can see this is a controlled vocabulary. You can only add anatomical structures, as I said earlier. Click the checkbox and the plus, and then it's added here automatically. You can see it's colored differently because you added it manually. All right, so we went over how you perform the registration, but now we actually have to discuss what you do with the data, how you get the registration data out of it. So if you use the standalone version of the RUI, which I'm demonstrating here, you can click this review and download button here in the bottom corner. And then you get this registration review window where we see all of the data that we just generated. So we see the first and last name of, me, the operator. Then we have the reference organ name. We have some geometric information, the dimensions, position, and rotation. Then we have these anatomical structure tags. You may want to double check these before you save the data out. A timestamp and an alignment ID, which is just a unique identifier. So then we click download registration data. I'm going to download this here to my machine. And then I suggest, let's just have a look at it. So I'm going to open this here in a code editor. Okay, so this is what the data that we just generated looks like. We have a whole bunch of boilerplate information up here. Then we have the creator first name and last name. We have the date. We saw all of that just a second ago in the review window. These CCF annotations correspond to the anatomical structure tags, except now here we have strings that represent URLs. Okay, here we have the dimension, which is 10 by 10 by 10 millimeters. And then here we have the placement object. And then this object, we have the scaling, we have the rotation, and we have the translation that corresponds to the 3D position that we set earlier. All right, that looks great. So there's another really neat thing that you can do with this JSON object that we just exported, and that is we can reload it into another instance of the RUI. So I'm gonna click this. I'm gonna refresh my browser, which of course deletes everything that we just did, because again, it runs client side in the browser. So, but I want to, let's say I want to edit an existing registration, or I want to just work with an existing registration, change it slightly and create a template that way. So I can type in my name again. I go to my organ and I choose the donor sex, left kidney. And now you can see all of this is just as it was at the beginning of our last experiment, where nothing is rotated, nothing is repositioned. But now we can click this upload previous registration data button. And then I navigate here to where I saved the registration data earlier. I click open. And then, like with magic, the tissue block was reset here into the middle where we had it earlier. And this way you can beautifully use these JSON files as templates for future registrations. All right. If you check this information button up here on the right, you get information about the RUI itself. You also get some information here about the background, what it has to do with HubMap. We link to a standard operating procedure, and there are also acknowledgements in here. All right, in this tutorial, I showed you how to use the registration user interface to perform a tissue registration for one of our many 3D reference organs. And I hope that you enjoy using the tool as much as we enjoyed it making it. Thank you.
It's really awesome to be able to look back and say, we've started with this group of people who have never worked together before 4 years ago. And we've now formed working groups. We've formed functional groups that are working on data portals.
Probably the major accomplishment of HubMap is just bringing this consortia worth of people together. I'm a biologist, but I am not a computational scientist or a data analyst, or I know what I want something to look like maybe on a website, but I certainly can't do that.
Yeah, so my main achievement, I think, is the validation system for the ASCT plus B tables. And I think that that system prompted me to think about how we could make more accessible views of the ontology products we build.
I think when I last looked, and this might have been last week, we had over 50 papers on bioRxiv for HubMap, and it's pretty awesome to be able to watch some of these folks just churn these papers out.
One of the big challenges is just collecting the data that we really need to now integrate all those data together to build the reference maps that we're looking to build that connect the molecular information with the spatial information across all these different types of molecules and assays. And that's a socio-technical challenge of setting expectations, of establishing standards in the consortium, and then doing it in a way that is win-win for everyone involved.
You're integrating data from many small labs. Other projects I've worked on, the standards have kind of emerged because of the dominance of very big institutions. And I think there's that balance between, like, I've got a minimal viable product and this is what I'm going to do in the next cycle of 4 months, versus I'm building a sustainable structure that is extensible and is going to be able to do all this integration work to build these cool tools. But every time that has to be focused on really clearly stated use cases, otherwise I think you're sunk.
There were certainly a lot of things that turned out as we expected in terms of data types that we could generate well. We could get lots of data, but could we also get groups who weren't necessarily the developers of the technology to also generate these data types? And I think we were pretty successful with that. We also ended up developing a number of new technologies along the way, which I think will be important for the next phase of HubMap. The next 4 years of the program will be what we describe as being the production phase. So we'll prioritize generating data types that we know we can generate well, that we know how to integrate them into some model of the human body. And then the fourth phase is really in the last year of the program, which is how we think about transitioning to some kind of sustainability.
I do want to see it more accessible, both for education and just for the person that's coming in because they're interested with this particular cell and they want to know how it changed from organ to organ or age to age.
As we're here at this demo day at the end of the first phase of the project, I see we're making progress towards that goal of starting to align with the work that Kadi and Rahul are doing to establish the framework for building these reference maps and connecting the data together in a way that will enable us to do that.
And something that really shows a proper linked open data approach where you can go very rapidly from I want to query the semantics, I want to query the metadata, to this is giving me a piece of data that I can do useful work with. I think that's the dream. Right now I'm going to sort of find data sets and then now I've got to mess around with how do I actually get to the data? Do I write to the person? Do I follow a link which then tells me to follow another link which then might end up at a dead end? Do I have a license? All of that stuff, that can be made frictionless.
So my dream in this time is to be able to establish a sustainable framework by which not only future projects that may follow HubMap, but also other projects throughout the world and other consortia that are working on similar problems can follow to one day, have that full, complete map of the human body at cellular resolution. That's not going to happen in 4 years.
I get the cool opportunity to be able to brag about all of the really awesome work that the scientists and the researchers and the technologists are doing. They're building pipelines and they're putting all of these really awesome tools together, and I get to tell everybody about it, my team, and that's really fun.
So how it works is that on the NIH side, we lay out what our overall goal for a program is. So for each of our 20 or so common fund programs, we have a stated goal. So for example, for HubMap, it's to establish the framework for mapping the human body at a single cell resolution. So we laid out the program essentially in 4 phases when we started. So we knew we had 8 years of funding to work with for this particular program. So we said, the first year is the year we're going to get everything set up, we're going to get it running, we're going to see how well we can generate data, make sure we can collect tissues, make sure that we have all the quality control checks in place that we need. So that was really the first year of the program, was that kind of setup phase. And then the second, third, and fourth years, we described as being the scale-up phase. So this was really the first 4 years were about trying different things to see what worked, see what we could generate data on, see how we could think about integrating those data types together. So that was really the first 4 years. There were certainly a lot of things that turned out as we expected in terms of data types that we could generate well. We could get lots of data. Groups who do this work, clearly can generate lots of data, but could we also get groups who weren't necessarily the developers of the technology to also generate these data types? And I think we were pretty successful with that. We also ended up developing a number of new technologies along the way, which I think will be important for the next phase of HubMap. And then I think there were some things that we tried which didn't work as well. So it's not quite clear how we think about those moving forward. How we incorporate them or prioritize them compared to other types of data. But really, the next 4 years of the program will be what we described as being the production phase. So we'll prioritize generating data types that we know we can generate well, that we know how to integrate them into some model of the human body. And then the fourth phase is really in the last year of the program, which is how we think about transitioning to some kind of sustainability for all the products we generate as part of the program. Because, obviously we don't want to spend 8 years doing all of this work, developing a lot of different things, and then as soon as the money runs out, we cut everything off. So, we want to make sure the products that come out of this program are sustained in some way, either through other NIH programs, through international efforts that are related to this, or just being pursued by different institutes or centers at NIH in specific disease contexts.
Yeah, so the organ system that we're looking at is the human digestive system, specifically your small intestine, your colon. So you probably know that when you eat food, it goes down your esophagus and into your stomach where it's very acidic and it gets a first level of breaking down. And then the material actually moves into your intestine, and your intestine's really in your colon, and those are two different components. So I have some analogies here. Your intestine is, first it's your small intestine, after food goes in the stomach, goes in the small intestine, and that's like this hot dog here. But imagine a 20-foot hot dog. That's how big your small intestine is. It's 20 feet long. And so the material actually passes down. There's like a tube in there that you'll be going through. And what happens is that will absorb nutrients which will pour into your body. And then from there it passes into your colon. And for this I've got a kielbasa. So you'll go from your small intestine into your colon. And same thing, but the colon has a somewhat different job. The colon actually is like a fermentation chamber. It will take a lot of the food, fiber you eat actually winds up in your colon and slowly gets broken down. And your colon has some very, very important parts to it that are just incredibly valuable to you. We tend to think icky when we think about colon and rectum and things like that, but your colon actually has bacteria, something called your microbiome. and it makes essential nutrients for us. It breaks down fiber and things that are important for us, but also makes critical vitamins, things like vitamin B12, things you may have heard of, that are actually really, really powerful for your health. I mean, you have to have these vitamins. That's why they're called vitamins. They're vital. And so your colon actually takes care of that. So the bottom line is we're going to make a map of basically this 20-foot hot dog along with a 5-foot kielbasa. And we're going to make a map at incredibly single cell resolution, very, very tiny level. We're going to figure out as best as possible where all the cells are, where as many molecules like RNA and proteins all map to within this digestive system. And so that's our mission.
This number of glomeruli.
I'm recording a video for work.
Referred to as calling cards. Nope.
And Connie will have a better answer, so.
Add to these tables.
Can I ask the final question?
You said the previous one was the final one.
I was introduced as always the next question.
All right.
Ready?
Yeah. HubMap will create. Can you do again?
Now you're in the frame. You're in the frame.
I was trying to think of something funny to say, but I'm very unfunny.
Creator uploaded.
Into that kind of exciting time where we can really brag about it.
Okay. So on this dark desert highway, cool wind in my hair.
I have one I realized we never got to the hot dog though. We have to back up at some point.
This one is a UMAP plot from the salmon RNA-seq thingamabob.
And that Richard would go with it.
This could either be an outtake or it goes into the movie.